Hello, everyone. Do you know that data of low quality leads to erroneous conclusions and analysis? That's unfortunately true, as it has been shown by many studies across the different modalities of MRI. This renders quality control, which is the process of vetting the quality of your data, crucial for the reliability of the study results. However, quality control is also time-consuming and highly variable because in the absence of consensus on systematic approaches to QC, the latter varies a lot between labs and even between researchers in the same lab. And such, we need appropriate QC protocols and tools to make QC more consistent, but also to make it widely adopted by lowering the barrier to implement it. In this process of vetting the quality of your data, in the end, you have to mark a trade-off between limiting the biases on your result, because as I told you, if you let in the analysis data of low quality, you have the risk of biasing your results. But on the other side of the trade-off, you also have to limit the amount of data that you throw away, because data are costly to acquire and you do not want to waste resources. So in other words, when you vet the quality of your data, you want to mark a trade-off between implementing a quality control which is too lenient versus too stringent. And to mark a good trade-off, we would like to highlight general guidelines that are easily implementable with existing tools, but have a sure effect of improving the reproducibility of your results. So the first guideline is to predefine your exclusion criteria before you actually do the quality control. And it is very important that those exclusion criteria are tailored to the particularities of the project. Consideration that have an impact on which exclusion criteria you define are research questions, planned analysis, types of data available, for example. The second guideline is that it is important to set up several checkpoints along the workflow. So this allows you to not only assess the quality of the preprocessing step, but it also offers an occasion to look at your data with a different perspective. And this overall ensures the robustness of the whole quality control protocol. To formalize this in a document, you should write a standard operating procedure, or SOP, which is a list of activity that you need to do in order to complete a task. And in quality control, this translates into writing a detailed checklist of all the steps you have to do to vet the quality of your data. To ease the establishment of such a protocol, we have developed a GitHub repository template called MRIQC SOPs. And this template on GitHub has all the mechanism to easily develop protocol. And don't be confused by the name. You can develop any type of protocol with MRIQC SOPs template. It doesn't have to be related to MRIQC, nor it has to be related to MRI. And this talk is part of a research topic that was organized, and the organizer asked researchers that are working on quality control to demonstrate their quality control on an example dataset and to give this protocol in many details. And so we wrote a protocol for this project using the MRIQC SOPs template, and let me show you how it looks. So this is the protocol that we have developed with the MRIQC SOPs template. It's written in Markdown, but you can easily generate an HTML page of your protocol. And as I told you, it's a checklist of all the steps you have to do. And also, I told you earlier that it is very important to predefine the exclusion criteria before you perform quality control. And as such, there is a page dedicated in the protocol to exclusion criteria where we give an exhaustive list of all the exclusion criteria that we have defined for the research topic project. And for each exclusion criteria, we give a thorough description of the criteria. So now you know that you should formalize your procedure in a standard operating procedure. Let me tell you exactly what we would like you to put as an ideal QC workflow in this standard operating procedure. So as I told you first, you define your standard operating procedure, which will govern everything like the acquisition, the pre-processing of your data, and how you will assess the quality of your data. And in there, I have told you also, you need to define your exclusion criteria that are tailored to the specific goals, type of data available, and etc. that is available in this project. Then once your SOP is written, you can move forward with the acquisition. So you acquire your data, and the first step then is to assess the quality of the unprocessed data. So in the context of a functional MRI analysis, you would probably be interested in thoroughly vetting the quality of the functional MRI signal, because that's your signal of interest. But you also might be interested in looking at the quality of T1 weighted images, because the latter will be used for co-registration or normalization, for example. So based on visualizing your data, you verify whether the subject fall under the exclusion criteria that you have defined in your standard operating procedure. And if they do, you have to exclude them. But if they don't, if they are of good quality, it's all good, you can move forward. And here I would like to distinguish two different aspects of vetting the quality of the data. The first aspect I would like to highlight is quality control. So you have a given set of data and you eliminate the subject so that they do not bias your results. But in an ideal workflow, I would like to see that you vet the quality of your data early enough in the acquisition phase, and you can use the knowledge from the quality check to inform your acquisition and improve the overall performance of this acquisition and avoid systematic biases and artifacts to spread throughout the whole dataset. And this is a process called quality assessment, where you have a feedback loop where you use the knowledge from the quality assessment to improve your acquisition. So once you have a tailored acquisition and you have a good quality subject, you can move forward and you can pre-process the data. And it's the same concept, you pre-process them, you inspect their quality using some visual report, for example, and based on the exclusion criteria that you have defined in your standard operating procedure, if the subject fall in one of those criteria, you have to exclude it. Otherwise, you can move forward, and so on, and so on, and so on. And for this research topic project, we have defined a specific quality control protocol. And let me show you to you right now. So it's focusing on quality control because the data were already acquired. So we cannot influence acquisition anymore. Then it's focusing on two quality checkpoints, the one on unprocessed data and the one on pre-processed data. Thirdly, I told you it is very important to have your exclusion criteria tailored to the particularities of the project and in particular to the planned analysis. So we define the planned analysis as whole brain voxel-wise analysis of spatially standardized resting state, bold fMRI. And lastly, we are implementing the quality checkpoints using the tools MRIQC and fMRIPrep. So now let me introduce the tools. So MRIQC is a quality control tool, and it generates visual reports that are depending on the modality. As of now, functional MRI and T1 weighted images were supported by MRIQC, but I have a cool announcement I would like to slide in this presentation, which is that we released a new version of MRIQC recently, and that new version supports diffusion MRI. So that's the manual aspect of quality control that is implemented with MRIQC. But MRIQC not only generates visual reports, it also generates image quality metrics. So those are metrics that are taught to reflect certain aspects of quality from image. And using those image quality metrics, you can implement automatic quality control. I will not talk too much about automatic quality control in this talk. However, last year at ICMRM, there was a full educational course fully dedicated on how to automate quality control. And they have developed a series of notebooks that are educational in the purpose of teaching people how to perform automatic quality control. So I will let you have a look. Don't hesitate to ask the community if you have any questions. Then the second tool that we are using is fMRIPrep. It's a pre-processing pipeline. So it outputs pre-processed data. It also outputs visual reports to assess the quality of those pre-processed data. And it outputs also conforms that you can use in subsequent denoising. Both fMRIPrep and MRIQC are pipelines that consist of a combination of tools from well-known software such as AFNI and ANS, and they are both designed to be open source, easily accessible. They require minimal user input. They are modular, and they are robust to variations in acquisition protocol. To execute MRIQC and fMRIPrep is thoroughly documented either on the NAPREP's website or on the respective tool documentation. So I will not go in details here. I will let you go through those resources. But please don't hesitate if you don't understand to ask any questions. I will give you my email in the last slide. And so now you know what we would like to see as an ideal quality control protocol. And I showed you guidelines. So let me make those guidelines more concrete by showing you examples of subject visual reports. So the first guideline I would like to talk about is the one about exclusion criteria that are tailored to particularities of the project. And specifically, let's have a look at this subject. So MRIQC visual reports looks like this. It's a collection of visualization, and they are designed to make you efficiently assess the quality of your data. And the one I want to show you for this subject is this one, the background noise, because the background is very informative to assess the quality of your data. And as such, let's have a look at this visual report. And what I want to show you is here. Here you have a wraparound, and the wraparound is the consequence of the field of view being too narrow, meaning the part of the brain that was supposed to be on this side gets reflected on the other side of the image. And here you can see that the wraparound overlaps with the brain. So let's imagine a scenario where you are studying decision making. Then you might be very interested in looking into what's happening in the prefrontal cortex. And as such, the observation of such a wraparound would grant exclusion of this subject. However, let's imagine another scenario where you are interested maybe at looking at the motor cortex. Then you might not care about what's happening in the prefrontal cortex, and as such, the observation of such a wraparound would not grant exclusion of this subject. So that's an example of why the research questions informs the region where data quality is important. Let me show you now why the planned analysis has an impact on your exclusion criteria. So now we are looking at the T1 weighted report generated by MRIQC. And the first visualization is a zoomed in view on the T1 weighted image. And the artifact I would like you to pay attention to is those motion related ringing. So in the case of this research topic project, we did not exclude subjects that present such mild artifacts of motion-related ringing, because the T1 weighted images are used only for normalization or co-registration, and those two preprocessing steps are relatively robust to such mild artifacts. However, let's imagine the other scenario where you want to analyze your fMRI signal on the surfaces. Then the surface that you reconstruct from these T1 weighted images will be compromised by those motion-related ringing. And as such, those ringing will grant exclusion of this subject. So that's a second example of exclusion criteria depending on the particularities of the project. Let me tell you now why the types of data that are available have an influence on the exclusion criteria. So we are back in a fMRI report that is also generated by MRIQC. And what I want to show you here is in the bold average. So it's the average of the bold across time. And what I want you to notice here is this signal dropout that appears in the ventromedial part of the prefrontal cortex, but also next to the ear cavities. This signal dropout typically appears next to brain-air interfaces, and they are due to susceptibility distortion. And in the research topic, because no field maps were shared with the dataset, this led us to exclude a substantial amount of subjects based on the observation of such susceptibility distortion artifacts. However, if you have field maps, you would not exclude subjects because you observe this artifact in the MRIQC visual report, because this artifact can be corrected by the susceptibility distortion correction implemented by fMRIPrep. And so you would delay the observation, you would delay the exclusion of your subject after fMRIPrep has run. But then, if after fMRIPrep has been run, you still look and you still see those residual susceptibility distortion, then you will exclude the subject. And this leads me to the next point, which is it is very important to set up several quality control checkpoints. And also you need your exclusion criteria to depend on where in the workflow you are standing. So let me now move on to the fMRIPrep visual reports, because we are considering the second quality checkpoints. And I want to look at this subject. So fMRIPrep visual report look similar to MRIQC. It's a collection of visualization, and each visualization is designed to help you assess the quality of one preprocessing step. So it's a collection of visualization, and the one I want to focus on here is the one on confound extraction. And particularly, I want to have a look at the confound extracted, which is called temporal COMCore. This temporal COMCore is a mask of the most variable voxels that are in the brain. And so typically, the most variable voxels appear in the outer rim of the brain, and also close to the brainstem, because those regions are filled with blood vessels and fluids. So you expect your mask to look scattered with individual voxels a bit everywhere. However, in this subject, you can clearly see here that the mask has a suspicious shape that reflects the presence of an artifact. And so, based on the observation of this mask, we excluded the subject. But it's a bit puzzling, because this artifact seems to be quite strong, and it is possible that we would have seen it in MRIQC visual report. So let's have a look at the same subject, but in MRIQC. And so we go through, and the one I want to talk to you about is the standard deviation map. It presents the standard deviation across times of all the voxels, and you have a mosaic presenting different axial cuts. The first one axial plane and the other one sagittal plane. And what I want to highlight here is those vertical strikes in the sagittal plane that show bands of high variability that are unrelated to physiological signals. And actually, this observation was actually an exclusion criteria that we had defined for MRIQC. So you see why having several checkpoints is extremely valuable, because it allowed us not only to double check our data, but to double check them with a different perspective that made the artifact maybe appear a bit clearer. So you see how overall having several quality control checkpoints make the whole quality control protocol more robust, and also the robustness of the MRIQC, the quality control protocol relies on the quality control to be dynamic, meaning you want to learn from different modalities, you want to learn from different visual reports, and as such, you can build a more coherent quality control protocol over time. So yes, that's all I wanted to tell you about quality control for today in this talk. The topic is really vast and I couldn't address everything in this talk, but I invite you to read our paper, in which we are giving an exhaustive list of all the exclusion criteria that we defined in the context of this research topic project. And additionally, in the supplementary material of this paper, we are illustrating how every exclusion criteria looks like on the visual reports of MRIQC and fMRIPrep. Also, please come and talk to us at the poster sessions. We have 3 posters related to quality control. The first one is about QK, which is a tool to which is a manual ratings manager to efficiently collect manual ratings from large datasets. The second poster is about leveraging wavelet transform to enhance quality control, specifically detecting B1 field dynamics. And the third poster is an example on how to analyze IQMs. And specifically, we show that SNR-derived metrics are predicting the presence or the absence of motion in T1 weighted images. I want to thank you so much for your attention. If you have any question, please don't hesitate to email me at this email address. And here I leave you all the quality control resources that I mentioned in the talk. Thank you so much.